तो इस लेक्चर में क्या पढ़ने वाले हैं हम? तो इस लेक्चर में हम डीएनए के बारे में पढ़ेंगे। इसके बाद इस टॉपिक से रिलेटेड प्रैक्टिस क्वेश्चंस भी हम डिस्कस करेंगे। तो आइए इस लेक्चर की शुरुआत करते हैं। तो हम क्या पढ़ने वाले हैं? विल टॉक अबाउट स्ट्रक्चर ऑफ द न्यूक्लियोटाइड चेन। ठीक है, नाउ लेट अस recapitulate the chemical structure of polynucleotide chain. It can be either DNA or it can be either RNA. ठीक है, तो जैसे डीएनए है, किसका बना है? न्यूक्लियोटाइड्स का बना होता है। इट इज अ पॉलीन्यूक्लियोटाइड्स, यानी कि न्यूक्लियोटाइड्स हैं वो आपस में ज्वाइंट होते हैं, उनके बीच में बॉन्ड होता है। ठीक है, कौन सा बॉन्ड होता है? फॉस्फोडाइस्टर बॉन्ड। नाउ अ न्यूक्लियोटाइड हैज थ्री कंपोनेंट। न्यूक्लियोटाइड के बारे में हम देखते हैं, न्यूक्लियोटाइड के कंपोनेंट्स क्या क्या होते हैं, क्योंकि इन न्यूक्लियोटाइड्स को मिला के ही क्या बनता है? डीएनए बनता है। नाउ अ न्यूक्लियोटाइड हैज थ्री कंपोनेंट्स। पहला कंपोनेंट क्या है? नाइट्रोजनस बेस। दूसरा कंपोनेंट है पेंटोज शुगर। तो कौन सी शुगर होती है? इसे पेंटोज शुगर होती है, मतलब कि उसमें 5 कार्बन्स होंगे। तो आरएनए के केस में इस पेंटोज शुगर को कहते हैं राइबोज, एंड इन केस ऑफ डीएनए this pentose sugar, this is called deoxyribose. So in both RNA and DNA ribose is present. Difference is that in RNA ribose is present, in DNA deoxyribose is present. So in both pentose sugar is present, means 5 carbon sugar is present. Now what is the meaning of deoxyribose? So in DNA you can see deoxyribose is present, which means at the second carbon the OH is not present, whereas in ribose which is present in the RNA, वहां पे जो सेकंड कार्बन है वहां पे OH is present. So this is the figure showing you difference between deoxyribose and ribose. Both are pentose sugar. Both have 5 carbons. What are the 5 carbons? Carbon number 1. This is the second carbon. Third carbon. Fourth carbon. And then this is the fifth carbon. Similarly in the ribose also, this is the first carbon. Then second carbon, third carbon, fourth carbon and fifth carbon. Difference is that in deoxyribose at the second position OH is missing, in ribose OH is present. Okay, so we were talking about the nucleotide. What are the components of the nucleotide? Nitrogenous base, pentose sugar and a phosphate group. Now there are two types of nitrogenous bases. So if we talk about these nitrogenous bases, there are two types of nitrogenous bases and they are known as purines and pyrimidines. Purines include what? Adenine and guanine, or you can say A and G, and pyrimidine it includes cytosine, uracil and thymine. So why we are writing uracil? Because RNA is consist of uracil instead of thymine. So purines which are A and G, they pair complementarity with the pyrimidines which are C, okay, cytosine, thymine or uracil, which means that there is a complementary base pairing between A and T. So A always pair with T, means adenine pairs with thymine, and there is a double bond between them, and G always pairs with C. Okay, guanine pairs with the cytosine, and there is a triple bond between them. So you can see it is difficult to break GC bond as compared to this AT bond, because AT it has 2 hydrogen bonds between them, and GC it has 3 hydrogen bonds between them. So this is the complementary base pairing. So in DNA, the 2 helix, they show the complementary base pairing between them. And what kind of bonds are present? Hydrogen bonds. क्वेश्चन डिस्कस करते हैं. What unique property does base pairing confer to the polynucleotide chain in the DNA? So we know that base pairing is seen in the DNA double helix. So what unique property does this base pairing give to the polynucleotide chains in the DNA? So DNA is made up of double helical structure. It is a double helical structure. Two DNA helix are present. Two chains are present. Now, what are the options? Option number A is they are identical to each other. Okay. Next is they are complementary to each other. Are the two polynucleotide chains, are they complementary to each other? Option number C, they are independent of each other. Option number D, they are randomly paired. Answer is option number B, means they are complementary to each other. Explanation देखते हैं. Base pairing confers the property of complementarity to the polynucleotide chains. तो बेस पेयरिंग की वजह से polynucleotide chains जो हैं they are complementary to each other. Now meaning if the sequence of bases in one strand is known, the sequence in other strand can be predicted because the other strand is complementary, okay, to the other strand. So both the strand they are complementary to each other. So if you know the sequence of one strand, okay, you can know the sequence of the other strands also. So this is due to complementarity of the two nucleotide chains, polynucleotide chains. And A pairs with T and G pairs with C. Now let's have a look at the incorrect option explanation. So option number A, this is incorrect because the strands are not identical, they are complementary to each other. Then option number C, the strands are not independent, they are complementary and they are interdependent. Option number D is also incorrect. The base pairs are not randomly paired. They follow specific pairing rules. A pairs with T and G will pair with the C. Now cytosine is common for both DNA and RNA and thymine is present in DNA. So cytosine is present in both DNA and RNA, but thymine is only present in the DNA. In RNA, instead of thymine, uracil is present. Now uracil is present in RNA at the place of thymine. Now a nitrogenous base is linked to the OH of 1 prime carbon pentose sugar through an glycosidic linkage to form a nucleoside. Now let's talk about nucleoside and nucleotide. So you can see there is a glycosidic linkage between nitrogenous base and the pentose sugar. So a nitrogenous base is linked to the OH of the first prime carbon, okay, of the pentose sugar. Means first prime carbon means carbon present at the first position. So there is a bond between them and this bond is known as glycosidic linkage. So you can say the nitrogenous base is linked to the pentose sugar with the help of glycosidic linkage. And this forms a nucleoside. So what is a nucleoside? You can see in this figure, nucleoside just consists of the pentose sugar and the nitrogenous base, which are joined together by an glycosidic linkage. And when you add phosphate with the nucleoside, then you get the nucleotide. So what is nucleotide? Nucleoside plus phosphate. This makes the nucleotide. The only difference is that of the phosphate only. Nucleoside doesn't consist of this phosphate, nucleotide consists of this phosphate. So here you can see a nitrogenous base is linked to the OH of the 1' pentose sugar through N-glycosidic linkage to form a nucleoside such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, then cytidine or deoxycytidine, and uridine or deoxythymidine. So we are talking about RNA and DNA here. So RNA has ribose sugar and DNA has deoxyribose sugar. So the pentose sugar is different in case of RNA and DNA. We have already discussed RNA has ribose, DNA has deoxyribose. So that's why we are writing adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. Now when a phosphate group is linked to the OH of the 5' carbon, you can see OH of the carbon which is at the 5th position of a nucleoside through phosphoester linkage. So the phosphate group you can see is linked to the 5' carbon of nucleoside through a phosphoester linkage. A corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Clear? So what is the bond between phosphate group and this nucleoside? It is a phosphoester bond, or you can say phosphate group is linked to the OH of the 5' of a nucleoside through phosphoester linkage. And when this happens, then a nucleotide is formed or deoxynucleotide can be formed depending on the sugar which is present. So this figure we have already discussed. This is showing the structure of nucleotide and nucleoside. This is the nucleoside which consists of a base and the sugar only, and when you add phosphate to it, then a nucleotide is formed. And you can see phosphate is linked to this OH of the 5' carbon. Okay, and glycosidic was forming between the nitrogenous base and the 1' carbon of the pentose sugar. So at one side there is a glycosidic linkage, another side there is phosphoester bond or phosphoester linkage. क्वेश्चन डिस्कस कर लेते हैं अब. Which of the following statements is correct regarding nucleotides and their components? So we are talking about nucleotides here and what nucleotides are made up of. What are the option? Option number A is thymine is present in both DNA and RNA. Then option number B, uracil replaces cytosine in the RNA. Then option number C, a nucleoside consists of nitrogenous bases linked to a phosphate group. Does nucleoside consist of a phosphate group or not? And option number D, a nucleotide is formed when a phosphate group is linked to 5' carbon of a nucleoside, it is saying. So let's have a look. Option explanation देख लेते हैं इसकी. A nucleotide is formed when a phosphate group is linked to 5' carbon of the nucleoside through a phosphoester linkage. So this is correct definition of a nucleotide which consists of nitrogenous base, a pentose sugar and a phosphate group. इनके ऑप्शन की एक्सप्लेनेशन भी देख लेते हैं. ऑप्शन नंबर ए क्या है? दिस इज इनकरेक्ट. थाइमीन इज प्रेजेंट इन डीएनए बट इट इज रिप्लेस्ड बाय यूरेसिल इन आरएनए. तो आरएनए में यूरेसिल होता है इंस्टेड ऑफ थाइमीन. ऑप्शन नंबर बी भी इनकरेक्ट है. यूरेसिल रिप्लेसेस थाइमीन इन आरएनए, नॉट साइटोसिन. So cytosine is present in both DNA and RNA. So it is common to both DNA and RNA. Option number C भी incorrect है. Nucleoside consists of nitrogenous base linked to a pentose sugar through N-glycosidic linkage, not a phosphate group. So nucleoside lacks a phosphate group. When you add phosphate to a nucleoside, then it becomes a nucleotide or deoxynucleotide, depending on the pentose sugar which is present. Now the two nucleotides, how the two nucleotides are linked now to form a polynucleotide. So two nucleotides are linked through 3,5-phosphodiester linkage to form a dinucleotide. So between two nucleotides, a dinucleotide is made up of two nucleotides. So two nucleotides, they are linked together with the help of 3' to 5' phosphodiester linkage. What does this mean? So this means that the 3' carbon of one nucleotide sugar is joined to the 5' carbon of the next nucleotide sugar through a phosphate group. So this creates a covalent bond between the two nucleotides, specifically called a phosphodiester bond. So you can see here the 3' carbon, this is the 3' carbon here, okay, of the one nucleotide. This joins to the 5' carbon of the next nucleotide through a phosphate group. So another, there will be a one more nucleotide adjacent to this. So this 3' carbon will link with the 5' carbon of the next nucleotide, of the adjacent nucleotide, through a phosphate group. You can see in the nucleotide at one end there is a 3' end and another one is the 5' end. So 5' end consists of the 5' phosphate here, 3' end consists of this 3' hydroxyl. Clear? So that's how the phosphodiester is formed between the 3' carbon of one nucleotide with the 5' carbon of the next nucleotide. And that's why it is called 3' to 5' phosphodiester linkage. Now more nucleotides can be joined in such a manner to form a polynucleotide chain. So more nucleotides can join together like in this manner by the phosphodiester linkage, and a polynucleotide chain is formed. So in the figure they are showing you a polynucleotide chain. At one end you can see 5'. You can say this is the 5' end where 5' phosphate is present, and here you can see this is the 3' end where you can see this hydroxyl. Now a polymer thus formed has at one end a free phosphate moiety. This is the free phosphate moiety at the 5' end of the sugar. Where is this phosphate present? It is present at the 5' end of the sugar, the pentose sugar. And you can see here what is happening. These are the nucleotides. Okay, nucleotide consists of what? Base. Adenine is the base here. This is the pentose sugar, and then you have the phosphate. Then again, this is another nucleotide here. This is the thymine is the base. This is the sugar, pentose sugar. This is the phosphate. Again, this is the guanine. Here, this is the sugar, pentose sugar. This is the phosphate. Again, here is the cytosine. This is the pentose sugar, and this is the phosphate. So here you can see at the end, the polynucleotide. At one of the end of the polynucleotide, you can see there is a 3' hydroxyl, and other end there is a 5' phosphate. All right. क्वेश्चन डिस्कस कर लेते हैं. व्हाट इज फाउंड व्हेन मोर न्यूक्लियोटाइड्स आर जॉइंड इन अ 3' to 5' phosphodiester linkage? What are the options? Option number A is mononucleotide, B is dinucleotide, C is polynucleotide chain, and D is nucleoside. Polynucleotide chain, this is the right answer. Explanation देखते हैं. When more nucleotides are joined together through 3' to 5' phosphodiester linkages, a polynucleotide chain is formed. When many nucleotides are added through phosphodiester bond, then what happens? A polynucleotide chain is formed. So this is the basic structure of nucleic acid like DNA and RNA. So DNA and RNA, they are the nucleic acids, and DNA is a genetic material now. Earlier RNA was a genetic material. Now what happens? So in this process what happens? The 3' hydroxyl group of one nucleotide forms a covalent bond with the 5' phosphate group of the next nucleotide. What is this covalent bond? This is the phosphodiester bond. So 3' to 5' phosphodiester bond is formed between the 3' hydroxyl of one nucleotide and the 5' phosphate of the next nucleotide. Then this linkage is called phosphodiester bond because it involves phosphate group bridging the two sugar molecules. And then you can see third step, as more nucleotides are added in this manner, the chain grows longer, forming a polynucleotide. इनके ऑप्शन के एक्सप्लेनेशन में देख लेते हैं. ऑप्शन नंबर ए जो है that is incorrect. Mononucleotide. So mononucleotide consists of a single nucleotide unit. सिर्फ एक ही न्यूक्लियोटाइड वहां पे प्रेजेंट होगा, which includes a nitrogenous base, a sugar, which can be ribose or deoxyribose, and one or more phosphate groups. It does not involve joining of multiple nucleotides. Now next option number B, this is also incorrect. Dinucleotide. So a dinucleotide is formed when only two nucleotides are joined by a single phosphodiester bond. While this does not involve 3' to 5' linkage, the question specifically asks how more nucleotides implying more than just two. तो dinucleotide कैसे बनता है? It is formed by two nucleotides which are joined by a single phosphodiester bond. Now next option number D, nucleoside. So nucleoside क्या होता है? It consists of nitrogenous base and which is joined with the sugar by a glycosidic linkage. So nucleoside consists of a nitrogenous base covalently attached to the sugar, which can be ribose or deoxyribose, but lacks the phosphate group. So nucleoside doesn't consist of a phosphate group. It is the precursor to nucleotides, but does not involve phosphodiester linkages. So there is no phosphodiester linkage in case of nucleosides. Now polymer thus formed has at one end a free phosphate moiety at 5' end of the sugar. This we already know. So this is referred to as 5' end of the polynucleotide chain. So polynucleotide will have a 3' end and a 5' end. Similarly, at the other end of the polymer, the sugar has a free OH of the 3' carbon. तो 3' carbon पे OH group होता है, यानी कि hydroxyl group होता है, and this is referred to as 3' end of the polynucleotide chain. So there is one 3' end जहाँ पे OH present है, ठीक है. 3' end पे OH present है, यानी कि हाइड्रोक्सिल ग्रुप प्रेजेंट है. 5' end पे फॉस्फेट ग्रुप प्रेजेंट है. Now the backbone of a polynucleotide chain is formed due to sugar and the phosphates. You should remember this also. A backbone of a polynucleotide chain is formed due to sugar and phosphate. These two make up the backbone of the polynucleotide chain. Now the nitrogenous bases linked to sugar moiety project from the backbone. So nitrogenous bases which are linked to the sugar, by a glycosidic linkage. So these project from this backbone which is made up of sugar and the phosphate. In RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. So how is ribose different from deoxyribose? So in ribose, 2' OH is also present. In deoxyribose, this 2' OH is absent. Okay, that's why DNA is more stable also because of this, because it lacks the 2' OH. And DNA is less subjected to mutation. In RNA mutation rate is quite high. In DNA, you have thymine instead of uracil. This also makes DNA more stable. Also in RNA, the uracil is found at the place of thymine. So in RNA, instead of thymine, uracil is present. And what is uracil? Uracil is present in RNA. Okay. Now you can see here the 5-methyl uracil. This is another chemical name of thymine. So thymine is what? It is 5-methyl uracil. So in RNA, uracil was present. It got methylated. Methyl group got added in the uracil molecule, and then thymine was formed. So you can say by this also that RNA was formed earlier than DNA. So earlier there was RNA world. RNA was the genetic material. Now DNA is the genetic material. So from this observation you can see that thymine is nothing but 5-methyl uracil. So uracil was present before and it got methylated, and then after methylation uracil was transformed into thymine. Now DNA has thymine, and DNA is more stable also because of this. क्वेश्चन डिस्कस करते हैं. विच ऑफ द फॉलोइंग स्टेटमेंट अबाउट द स्ट्रक्चर ऑफ पॉलीन्यूक्लियोटाइड चेन्स इज नॉट करेक्ट, मींस विच इज फॉल्स. तो ऑप्शन देख लेते हैं. ऑप्शन नंबर ए क्या है? अ 5 प्राइम एंड ऑफ चेन has a free phosphate group. So the 5' end of the polynucleotide chain, does it have a free phosphate group? Option number B, the backbone of chain is formed by sugar and phosphate. Is it true or false? Option number C, in RNA, uracil replaces thymine found in DNA. So in RNA, uracil is present instead of thymine. Then option number D, the 3' end of the chain has a free phosphate group. Does 3' end has a phosphate group? Option number D is the right answer. The 3' end of the chain has free phosphate group. This is the right answer. Explanation देख लेते हैं. Statement that the 3' end of the chain has a free phosphate group. This is incorrect. The 3' end of the polynucleotide chain has a free OH group on the 3' carbon of the sugar, not a free phosphate group. Free phosphate group. Okay, the phosphate group is present at the 5' end of the polynucleotide chain. इनकरेक्ट ऑप्शन के एक्सप्लेनेशन भी देख लेते हैं. ऑप्शन नंबर ए जो है वो इनकरेक्ट है. द 5' end of the polynucleotide chain indeed has a free phosphate group as explicitly stated. Okay, as we have studied. Option number B, this is also incorrect here because the backbone of polynucleotide chain is formed by sugar and phosphate. Then option number C, in RNA, uracil replaces thymine found in the DNA. So in RNA, instead of thymine, uracil is present. So all the statement A, B, and C, they are correct. And the incorrect statement here is option number D. And this is the answer here. Although it is incorrect statement, but this is the answer here. Now DNA is an acidic substance. So DNA is acidic because it has a phosphate group which is negatively charged. And this is the reason for acidity of the DNA also. So in DNA, what is the full form of DNA? Deoxyribose nucleic acid. So you can say DNA is acidic in nature. It is an acidic substance which is present in the nucleus. So DNA is present where? In the nucleus. So this was first identified by Friedrich Mischer in 1869. Now he named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of the time. Now, it was only in 1953 that James Watson and Crick, based on the X-ray diffraction data produced by Morris, Wilson and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of the DNA. So Watson and Crick, they gave the double helix model for the structure of the DNA. Clear? We'll talk about this also in the upcoming slides. We'll talk about the double helical structure of the DNA. You already know about this double helical, a model of the DNA. क्वेश्चन डिस्कस करते हैं. What name did Frederick Mischer give to the substance he identified in the nucleus? What are the option? Option number A is DNA, B is nuclein, C is chromatin, D is helix. Nuclein is the right answer. Explanation देखते हैं. Frederick Mischer ने named the substance he identified in the nucleus as nuclein, and this was acidic in nature. Other options are incorrect because DNA is the modern name for the substance chromatin. Okay, for the substance chromatin refers to the complex of DNA and proteins in the nucleus. So what is chromatin? It is a complex of DNA as well as proteins which are present in the nucleus, and helix refers to shape of the DNA structure proposed by Watson and Crick. So they talked about helical structure of the DNA.